All right, what we're going to look at this time is going to focus on the process of RNA splicing. Once again, the idea behind splicing is to get rid of the intron sequences, glue the exons, or at least some of them together. Now, in different cell types, you may have what we call alternate splicing, where you will throw some exons out, as well as the introns, or in some cases, even parts of exons. We'll see some of those extra little complications later on. But the whole idea behind splicing is to get rid of the exon sequences, because of course they don't have the information you need to code for a protein. If you kept intron sequences in the final messenger RNA, what would happen is the ribosome wouldn't realize that that happens to be sequences, effectively commercials, so it would start adding amino acids based on the nucleotides in that, and you'd get a completely screwed up protein. At best, probably wouldn't work, and at worst could be down have harmful interactions that you're not aware of. So you gotta make sure you get rid of all those things. And once again, it's only when splicing and polyadenylation and at least partial capping are complete will that RNA ever be able to leave the nucleus. We'll see more on that after the test. Okay, well, splicing is done once again by this enormous dynamic complex called the spliceosome. And the spliceosome consists of what are called the small nuclear RNPs, RNA protein complexes, or SNRPs as people in the field like to call it. And for the spliceosome that deals with the polymerase II transcripts, in other words, the ones that are going to make messenger RNAs, that includes what's known as U1, which is the U1 RNA, a small one, about 150 nucleotides long. Some core proteins are common to almost all these SNRPs, and then a set of unique proteins. So you have U1, U2, U4, U5, and U6. U3 is involved in cutting parts out of ribosomal RNAs so you don't find it in the regular spliceosome. It's got its own role. And there's a whole bunch of other smaller, minor SNRPs that are used for specific RNA types and things like that. We don't have to worry about that. There's a whole bunch of others. Okay, well, we have the SNRPs. Each one of them. And then we have what are called general splicing factors, that are proteins that assemble and do things on the spliceosome and then generally leave. There are probably some that are also permanent components of it. And then we will also have specific splicing factors. And these specific splicing factors lead to certain types of splice pairs, like alternate splicing. One cell splices things in one way, another cell splices things in a different way. It's the same preliminary RNA precursor, yet spliced differently. What does that are these specific splicing factors. So in a sense, you can look at the spliceosome, and in some ways, it's kind of like the whole assembly that's involved in transcription. You got the common for transcription, you got this basal complex, that's sort of like your kind of core spliceosome, and then you have these very specific transcription factors that are going to give you cell-specific changes in transcription. So we have the same kind of thing. And like this whole transcription assembly, the spliceosome is an enormously large set of molecules, and things are coming in and coming out during the splicing process. So let's take a little look at that splicing process. Now, a second thing, too, before we get to that, once again, we have to recognize where an exon ends and an intron begins. We have to recognize the boundaries between introns and exons. So here's an exon. Here's an intron. Now this is an RNA. We have we have a couple things on the boundaries that are absolutely invariant. In other words, there are no differences in any intron-exon boundary that we see. So we have over here, we have an AG, 2 nucleotides. Over here we have a GU. Now there's other stuff here too that goes beyond that, but these sequences are invariant. People have done experiments with genetic engineering tricks. If you alter as much as a single nucleotide in those two ends, that completely abolishes splicing. You absolutely positively have to have these sequences. Now, nucleotides down here and down here are also going to play a role in splice site recognition, but they will vary. And some splice sites, as we're going to see, just like we mentioned about some promoters, there are what we call strong splice sites that promote spliceosome assembly, and there are weak, wimpy splice sites that make it less likely for spliceosome to assemble. We'll see that later on. Okay, so we have that. Now we also have something over here, it's a few tens of nucleotides away, called the branch point. And you'll see that, and a region here called the polypyrimidine tract. Now, pyrimidines are, there's purines and pyrimidines, I forgot which are which in terms of the bases of nucleic acids. Does anybody had in molecular biology remember that?
The pyrimidines?
Yeah.
Those are the thymine and...
Thymine and okay, uracil and cytosine, okay.
Yeah, yeah.
Okay, uracil and cytosine. Okay, all right, so uracil and cytosine. So polypyrimidine means it has a lot of pyrimides, lots of uracils and cytosines in that stretch, a few tens of nucleotides long. These sequences, although they vary from one intron to the next, there are some common features and they are also very important. So these are the critical sequences we see. The most important here, of course, are these invariant sequences. Get rid of those, alter those the slightest, and splicing is completely abolished there. And this sequence, as we'll see, is rather important. Okay, so now what we're going to do here is we're going to start the... Yes?
Before you read that, is the branching point before or after? Like,
I
real
think,
close,
now don't
like,
quote me on this, but I think it's actually inside, a little inside that polypyrimidine tract. Don't quote me on that one, though. I have to look that. I actually don't know for sure offhand, but I believe, ancient memories, I believe that the branch point's actually inside that. Or if not, it's darn close to it because they both have important roles to play. Okay, well, let's try to do the process of splicing. I'm going to do several different drawings. They're going to be a little bit crude, granted, but I'll give it my best shot. Okay, so what we're going to do here.
Quick question. We're still talking about regular splicing, not the alternates.
Yeah, we're getting the basics, how does splicing occur. We'll deal with alternate splicing a little bit later, how that works. We'll deal with that a little bit later. But this is generic. How does splicing work? How do spliceosomes work? Okay, once again, now what I'm going to do here is I'm going to stretch this boundary quite far out here. So I can put all the players in here. Okay. And I'm just going to erase a few of the labels. Okay, at the very beginning of what happens is we're going to have, in the first stage of splicing, we're going to have our U1 SNR bind to the AG and adjacent sequences. So it sticks to one part of the splice, one of the splice junctions. Now, around in that polypyrimidine tract close to the branch point are going to be a couple of general splicing factors. One's called U2AF, U2 auxiliary factor, and I forgot the name of the other one, but it's a couple proteins. These are general splicing factors. You need these splicing factors for any splicing to occur. And what happens is these two proteins not only bind to the appropriate part of the intron, they bind to each other, and now that acts as a docking point to get our U2 coming in. So now we have our U2, and the U2 binds a little bit, but it mostly binds these proteins. Now notice this U2 is not quite at that splice junction. It's close to it, but it's not quite there. All right. So we've assembled the first part of the spliceosome. Now the second part is we're going to put a complex of 3 of the remaining 3 SNRPs come in as a complex. And you have U4, U5, and U6 come in as a complex. What these things are going to do, at least at the initial phases, is act as a bridge between U1 and U2, causing things to kind of like bend out a little bit. In fact, I'm going to draw that over here. So it might be a little bit like this. We get a kind of loop. So we're joining U1 and U2 together. Now, we've kind of assembled the major characters and a few of the general splicing factors. Now what we're going to do is actually start to cut things. Okay, so our next step is the U1. Question?
I was trying to figure out in this book, and I got it.
Oh, okay.
You did.
Okay, it's the second set of illustrations in Figure 2, the process of splicing. Okay, now the next thing we're going to do here is we are going to cut right over here at that splice boundary. And U1 plays a role in that cutting. Exactly what it does and how it does that, we're not quite sure yet. Yes?
This cutting takes place after the U's have bent the intron.
Yeah. Yeah, after the major 5 SNRPs have started assembling together, now we start doing the cutting and the splicing reaction. First we have to get the characters into place. And then we have to start slicing and dicing. So, now what we're going to do is this. And this is going to be a little hard to describe. We're going to cut the RNA right over here. We still have to hold the exon in place. We can't let this exon be released. And now what we're going to do, and I'll kind of deal with this guy later. Now what we're going to do, we cut the RNA, and then this RNA part of the intron loops around and is covalently attached to that branch point. And this RNA in the form of a loop, which is the intron, is off. It looks like, I guess whoever first discovered this was a Westerner. He said, oh, this looks like the old lariats on the Wild Wild West. So they often call this the lariat RNA. Okay, now the second thing, of course, is we don't want to release this exon. So this whole spliceosome kind of reassembles itself, and this is going to be a little bit funky to try to draw. Let's see if I can draw this stuff in. All right. U2 is holding things. Yeah.
Okay, the second. Okay, U2 is right here, and then U2 is on this side as well. What is in the middle?
Oh, U2 is on this side? Oh.
Yeah, like right there.
Oh, those are some of those proteins, those general splicing factors that guide and stabilize U2. So they're not SNRPs, but they're splicing factors. Okay, that U2 auxiliary factor. Now, once again, I'm going to try to kind of redraw things because things shift around. And we are now going to have U1 seems to be able to bind to U2. U1 is still holding on to the exon. Okay? Because we don't want to let the two exons go. We have to hold the exons together. And then we're also going to have 4, 5, and 6 somewhere around here. So U4, U5, oops, and U6. Okay, so once again, 4, 5, and 6 are playing this bridging role between U1 and U2. U1 and U2 are now closely spaced together. U1 is still holding that exon in place. You don't want to lose that. All right. Now, the other thing is we have our lariat RNA. So the first step is cut. First step in splicing after we assemble things together. All right. Now we're actually going to have a rearrangement of the reassembly of the spliceosome. We're going to start getting rid of a few things, and we're still going to hold a couple things together. So our next step is going to be to rearrange the spliceosome, but now we're going to cut this boundary here too. So our next set of steps. And we have to join the exons together. So now what we're going to do is I'm going to have to redo this whole diagram once again. Sorry about this, but here we go. Okay, we have to cut. We're going to cut here. Okay? Question?
Yeah. So what we just did, is it separate from what you're about to do?
Yeah.
So it's
This is
separate.
sort of like the next step.
Okay.
Yeah. So the first step, step 1, assemble the spliceosome. We did that. Step 2 was cut the first intron-exon boundary. Form the lariat RNA and still hold the 2 exons in the complex. You don't have one drift off and lose it.
Okay.
Okay. Now the second step, which requires a big reorganization of the spliceosome in terms of positioning things. Some things leave, some other things enter. And the next step now is we have to cut that second exon. And we have to glue exon 1 and 2 together. And then the lariat RNA is a separate piece. So, cut, cut again, and splice. And that requires a big, yes?
Okay,
You're gonna draw
so
that.
this exon that you cut off over here, it's this exon.
Yeah, exactly.
Okay, so it's the same thing, you just moved
Right, I just moved it over there to show you that it's not drifting off. And U1 is still holding on to that exon. Because if you don't, it drifts off and then you're toast. Okay, so now we have to do this whole thing. Okay, we'll put, for clarity, we'll put exon 2. Now we have exon 1 here. Okay. What seems to be associated with those 2 exons is mostly U5. In fact, it's believed that U5 has the capability of doing that, what we would call ligation reaction. In fact, it probably also is the thing that cuts at the intron-exon boundary, the second exon. So remember, U2 doesn't stick to the boundary. It sticks close to it, but not at the boundary itself. So U5 seems to do a lot of the work. That second cutting, that second cleavage, and then the ligation, the joining things together. Yes?
So is there anything in between those two? Are they?
Not at this point. Well, I just put a little space to see that they're not
Okay,
separate.
but that like
Yeah,
thing.
we're just holding together and joining. Okay, so why don't we do this for clarity here? Okay. We just join them together. How about that? Okay, well, we've got that. Okay, now meanwhile, we have that lariat RNA that's going to be released. IO silver. Okay, here's your lariat RNA. Okay, actually I should flip it around to keep our orientation right. Okay, wow, like,
Oh.
God. I might say things that I'm not supposed to say in the classroom setting with this chart. Okay, all right. We're still going to have U2. Recall, U2 was at that branch point. U2 is still kind of holding us together. It also appears that U6 is associated with this. Meanwhile, here's our lariat. It's now separate. You notice it's separate from the two exons. Here's our lariat, and meanwhile, U1, its job done, and U4, its job also done. They hightail it out of here. They're not needed anymore. Okay, so the spliceosome is starting to disassemble at this point. Okay, now we have gotten the splicing reaction. We join the two exons together. So now we can finally get rid of U5. It drifts off, and later on, once again, later on, U4, 5, and 6 will join together to form a complex, and it's ready for the next splicing reaction. Okay, so now we got our exons. We don't need these other SNRPs anymore, so we have the lariat RNA left by its lonesome, and pretty soon it gets destroyed by nucleases. We don't need this stuff anymore. Don't forget that lariat RNA, that's the intron. We don't need that intron anymore. We got rid of it, just trash it. Okay, so there's three major kinds of, let's, without the drawings, let's kind of summarize those again.
What destroys the lariat RNA?
Lariat RNA, many nucleases that you're going to find inside the nucleus. There's all kinds of RNA digesting enzymes in the nucleus, which is why so little RNA that is transcribed actually makes it out of the nucleus. Most of the time it's stuck in there, and then bye-bye. Okay? All right, did that help?
Yeah.
Okay. Okay, our first set of steps is basically assembling the spliceosome. U1 goes to the AG at that first intron-exon boundary, sticks there. U2 and the splicing factors are needed to assemble, to hold U2 in place. That binds in that polypyrimidine tract very close to the branch point. And then finally, 4, 5, and 6 come together as this complex and serve as a bridge joining U1 and U2 together. So that's our first step. Our second set of reactions. Now we're going to cut U1, apparently it's U1 at least, cuts that first intron-exon boundary. Okay? So we could say this is the first cleavage or cutting reaction. Okay, major things in this. U1 is going to cut that at that AG, that first intron-exon boundary. U2 is going to help to join it. We don't know all the details of this, by the way, but it seems, since it's located in the right place, it probably is what actually does this. U2 is going to join that end of the intron. We just cut that thing off It's going to loop it around, we're going to join that end of the intron to the branch point. And that's where we make our lariat RNA. So now we've gotten the spliceosome, the two exons. They're separate from each other, but still held together in the spliceosome. Plus the lariat RNA, and that's attached to our second exon, and that thing is still being held in place. Okay, last but not least, the third set of reactions is where we do our second cut and then we join things together. And that's where you get our second exon cut, the joining of the two exons together. U5 is believed to do most or all of that. Okay, and then U2 and U4 are still bound to the lariat RNA. U1 and, or rather U2 and U6, U1 and U5, or U1 and U4 leave. And at this point, the splicing reaction is completed. Now we just get rid of all the SNRPs, and we're good to go again. All right, that's our basic process of splicing.